Namaste all of you. My name is Dr. Ritu Singh Rajput, Faculty of Agriculture and Veterinary Science, Department of Food and Biotech, JAYOTI VIDYAPEETH Women's University. In our previous lecture, we had discussed about the streptokinase and urokinase. Today's our session name is today's session name is recombinant coagulation factors. So, what is the recombinant coagulation factors? Hemostasis is the process forming clots in the blood walls, which is damage blood vessels and preventing blood loss, and while maintaining blood in the fluid state within the vascular system. And these are responsible for the blood coagulation and clotting. Coagulation or clotting is a process which blood loses its the fluidity and becomes the jelly-like mass in a few minutes after the shed. Now I will tell you about the history of the coagulation factor. In 1720, Jean-Louis Petit hemostasis was caused by the blood clots. After that, 1816, Rudolph described the blood clots. After that, the 1905, Paul Morwich assembled the four coagulation factors. In 1914, Paul Orphan also recognized some factors. What are the factors involved in the blood coagulation? Coagulation of blood occurs through the series of the reaction due to the activation of group substances. The substances are necessary for the clotting for called as a clotting factor, which is also called a coagulation factor. There are so many types of factors, which is factor number 1, which is called as the fibrinogen. Factor number 2 is a prothrombin. Factor number 3 is a thromboplastin, which is also a tissue factor. Factor number 4 is the calcium. Factor number 5 is the labile factor, which is the proacylene, and that is also acylate globulin. Factor number 6 is the presence has not been proved, and factor number 7 is the stable factor, and the factor number 8 is the anthrophilic anti-hemophilic factor, and the factor number 9 is called as the Christmas factor. Factor number 10 is called as the Stotter power factor. Factor number 11 is the plasma thromboplastin, and factor number 12 is the Hagman factor, and factor number 13 is the fibrin stabilizing factor. So there are about 13 total blood coagulation factor which is involved in a blood clottings. Their clotting factors are the protein which form the enzymes, and they are proenzyme and enzyme clot information, and that reaction involved into the conversion of proenzymes and enzyme are explained generally in the 3 stage. First is your formation of prothrombin activator. After that, the conversion of the prothrombin in a thrombin. After that, the conversion of fibrinogen into fibrin. So they can be easily, there are two types of pathway where work for the they are working for the sorry they are working for the blood clotting factor, which is in intrinsic pathway and extrinsic pathway. In intrinsic pathway, factor number 12 working, and extrinsic pathway there is related to the tissue factor. And intrinsic pathway first is the intrinsic pathway related to the heme factor, which is factor number 12, and it is activate the thrombin, and that after that it is activate to the R prothrombin, and it is converted the fibrinogen into fibrin factors. And in intrinsic pathway, this is related to the tissue injury, and it will be activate thrombin factor, which is factor number 8, and it is activate to the tissue factor number 8A. Then it will be activate our calcium. After that, it will be converted, it had to activate the thrombin, and thrombin converted into the fibrinogen, and fibrinogen convert into the fibrin protein. Then it can be easily clot to the our blood system. There are some feature of the intrinsic pathway and extrinsic pathway, which is in an intrinsic pathway, if so the first of all I will told you the source of the thromboplastin, which is damaged platelets in an intrinsic source. In extrinsic source are the damaged tissues. There is a platelet and there is a tissues. The role of factor number 7 is not required in intrinsic pathway, but in extrinsic pathway there is a required factor number 7. Number of reaction is more in intrinsic pathway, but number of reaction in less in extrinsic pathway. In intrinsic pathway the time duration very few minutes, but in intrinsic pathway it will be work in few seconds. It is activate from the factor number 12 and it is activate from the factor number 7 in the extrinsic pathway. Now we will discuss now I will tell you what is the generally all in our previous lecture we have discussed about the human growth hormone, insulin, facile stimulating hormones, erythroproteins, tissue plasminogen activators, factor number which is related to the essential blood clotting factors. So these also all are part of our clotting factors. It is essential blood clotting proteins which is also anti-hemophilic factors. This is called also as AHF. It is human factor that is involved. Factor number 8 is encoded by the F8 gene. This protein is circulated into the bloodstream within an inactivate form, and it can be easily bound on other molecules and called as a Wimborne factor. And it helps to injure that damaged blood vessels occur. So this factor is also helpful to the damage injury by the in the blood vessels, and it is work so fastly that is also called as a that is why it is called as a Wilbur factor, just because this is found at the Wilbur. These active proteins are interact within the another coagulations and factor number 11. This interaction sets of the chain of addiction chemical reaction, and easily it will be clot the blood. First of all, for example, I will tell you how it is work in simultaneously. First of all, if we get the, injury in our damaged and skin blood cells, after that it will be activate protein interact with the factor number 11, and it will be after that it will be chain reaction leading to the formation up to von Wimble factor. Then it will be activate factor number 8. Then factor number 8 related to the factor number 8 related to the intrinsic pathway. Then it will be activate the factor number 12, it is a part of the extrinsic pathway, and it will be activate to our blood clotting system easily. So this is all process how it is work easily in a proper manner. And F gene, which is related to the factor number 8, it is worked so properly, and some mutations such as a large of inversion described in above. I have told you how blood clots form, how it will be work in a hemophilia, and how it will be work in, first of all, factor number 8 is isolated by in 1919, which is produced in hamster cell, which is available in the hemophilia factor. Just because in hemophilia we had study about the hemophilia is a very our blood is simultaneously passed out from the our body, so we have to, if just because our blood is not clotting, there is no regulation blood coagulation factors work properly, so we have caused the hemophilia. So we have the factor number 8, which is RHF5, also called a factor number 8. So this is produced by the hamster cell, so it is help in a hemophilia hemophilia A for treatment. And this protein is produced by the Chinese hamster ovary and the baby hamster kidney cells. This factor is isolated by the Chinese hamster from the hamster kidney baby cells. And for first time it is RHF it is produced in a human has whose cell line available after the research how it is working, how it treated, and if in incorrect reproduction of the PTM in the non-human expression system can be triggered by the immune reaction. It is not properly treated by the particular factor. It can be not easily, it is easily coagulate in the blood, and it cannot be activate our fibrin, and it is not easily converted into the plasmin, into the plasminogen into the plasmin, and it can't be activate the our fibrin proteins. So how can we, it will be converted, it will be activated our coagulation factor. So inhibitors developed occurs that throughout the hemophilia patient. It will be helpful to particular constraint and healthcare systems. And there are so many factors we have studied about. We have got around 12 to 13 factors in our body. Some are identified, some are non-identified, which some are factors are usable, some are factors are activated, some are activated. And first of I will tell you how now I will tell you how to we purify, how to we produce these factors by the production. First of all, we have to harvest, then after that we have capture, after that we have to done a purification process, then we have to done DNA removal, then we have to activate virus inactivation, we have to done affinity production, after that we have done virus removal. So in harvesting we are use cell removal, in captures we have used multimodal cations, and after that we in purification we use cations exchange chromatography, which is column chromatography. After that, in our DNA removal we have used by the charge filtration. In virus inactivation we have used chemical inactivation, which can be solvent or detergent. And affinity purification we have got a factor number 8 selected by the chromatography columns, and after that virus removal can be done by the nanofiltration. So this is all about the recombinant coagulation factors. Thank you. Now I will tell you the examination point of view. They can be asked some question which can be how many factors are present in our body, how can we purify our factors, when they, how many types of pathway are present in recombinant protein factors, how can we produce these factors, how can we activate these factors. So exam point of view, these question will be asked in your examination. Thank you.